I'm Teresa Kozin, Executive Director at the California Wolf Center, and I'm pleased to have so many members, supporters, and volunteers joining us today. We are taping today's webinar, so people who cannot attend will receive a link of the program within the next week. As many of you know, the mission of California Wolf Center is to support the return of wild wolves to their natural habitat and to the people who share the landscape with them. We do this through conservation, education, and research. Today's webinar is about the fascinating subject of theriogenology. So we're picking up on our tenet of research for today. Theriogenology is veterinary reproductive medicine, and as it applies to the recovery of endangered Mexican gray wolves, which is very important to our mission at California Wolf Center. To help these wolves rebound from the brink of extinction, captive breeding programs such as ours at the California Wolf Center must work closely with those managing reintroduction of populations. Our guest speaker is Dr. Bruce Christensen. He is a respected authority within the elite field of theriogenology. He's been recognized for his clinical expertise in the canine breeding industry, as well as his academic achievements. Dr. Christensen founded Kokopelli in 2017 in Northern California to provide that area with specialized knowledge and skill in assisted reproduction in both canine and equine species. Dr. Christensen has a stellar academic degree background and career. He's been an active leader in the Mexican gray wolf species survival plan, or now known as a SAFE plan, by leading the scientific group of experts involved in managing the collection and banking of semen. We came to Dr. Christensen when he visited California Wolf Center in February of this year to manage a semen collection on one of our packs of Mexican gray wolves. It was truly a learning experience for all of us involved. As staff, we'd not been participated in a semen collection, so we're very happy to have Dr. Christensen there to work with us, to lead us, and make it a very educational experience. The moderator for today's webinar is Bree Harris, our development coordinator. She'll be taking questions for the speaker later in the program. Sarah McIsaac, our Wolf Care and Reintroduction Coordinator, is also on the webinar today. She'll help answer any questions regarding our involvement in recovery and reintroduction of the endangered Mexican gray wolf. Feel free to type your questions in the Q&A feature on the webinar. As Dr. Christensen is speaking, Bree will collect them. We'll have time for a Q&A after the presentation. And now, please welcome Dr. Christensen.
Hi, thank you for having me. Am I coming through fine?
You are, yeah.
Okay, great. All right, so I'm really pleased to be here, and I'm going to jump right into the topic. So as was mentioned, I am a veterinarian, and beyond that, I've specialized in reproduction and fertility, and in the veterinary world, that's known as theriogenology. So it's essentially the equivalent of an OBGYN and a urologist, all rolled in one, but for animals. And I got into this field, into veterinary field, because I was interested in zoo medicine and conservation medicine. And I ended up specializing in reproduction and fertility because I really felt drawn to the captive breeding programs in zoos and their contribution to endangered species. And so my work with the Mexican gray wolves has fallen right in line with that, and I'm really grateful to have been able to play a role. So I'm going to talk today. I'm going to share my screen now and get my presentation ready for you. Okay. All right. So the specific area that we're going to talk about today is the role of assisted reproductive techniques in the Mexican gray wolf survival plan. And my role in that is helping with semen freezing and artificial insemination in the captive population, which then augments the wild population. And there is some flow back and forth between, some pretty active flow back and forth between the wild population of packs that are surviving now, having been reintroduced in Arizona, New Mexico, and Mexico, and then the captive population. So we're going to talk about how we work with that. So freezing semen in Mexican gray wolves has been done since 1991. We began to collect and freeze and store semen back then, and so we have genetics from males that were alive 30 years ago, and that is, in theory, good for decades. And I can tell you that with domestic dogs, we have on occasion used frozen semen from domestic dogs that has been frozen for 30 years to breed other domestic dogs and gotten nice, viable, healthy litters of puppies. So the theory of being able to preserve genetics in perpetuity is very much in practice as well. But I'm going to talk today about what's happened specifically with Mexican wolves in that respect. In this picture, so let me give a quick disclaimer now that we're already into it, actually. There will be some pictures like this throughout the presentation of wolves that are under anesthesia and being worked on. There are no gory, bloody pictures in this presentation at all, but there will be pictures of wolves that are under general anesthesia and being worked on. And there's one picture of a wolf capture. So I want you to be aware of that as we head into it. In this picture here, there is a wolf on the table. You can only see its tail and its hind end because that's the end I'm working on. The front end is covered by people, but you can see up at the right-hand part of this picture, there's actually an anesthesia unit up here, and somebody is monitoring the anesthesia on that wolf to make sure that he's safe and doing well under the anesthesia while we're doing this project. Now, the way that we collect semen from wolves, they have to be under anesthesia. When I collect semen on a domestic dog, he's very much awake, and we just use manual stimulation, and he gets excited and voluntarily gives us an ejaculate. But you can imagine the obvious reasons why this is not something that would ever work with a wolf, certainly not a Mexican wolf that has been purposefully kept isolated from active contact with people in the hopes that some of them can be reintroduced to the wild. We don't want them to become habituated to positive contact with people. So they're under anesthesia, and the way that semen is collected is through very light electrical impulses given through the rectum around the area of the prostate, and that gently stimulates nerves in the hind end. You can tell that it's working because some of the nerves that are stimulated go to the muscles of the legs. And so when the process is happening, the legs will contract and flex just because of stimulation. When you have your muscles move in your body, there are light electrical stimulations that your brain and nerves are producing that have your muscles move, and this process mimics that. So we'll see the legs moving. We don't care about the legs. Mostly what we care about, well, all what we care about is the stimulation to the penis and to the nerves that control ejaculation. So what you can't see very well in this picture, but what's happening here, so here's the probe. It's about the size of your thumb that is in the rectum, and the electrical currents are being directed downward or toward the belly of the wolf. And then underneath this towel here is a leg, and you've just got a towel over it because the leg has dirt on it, and we don't want to get dirt in the sample. There's a surgical drape here just to keep things clean. It's not a sterile procedure, but we're trying to keep things clean. And there's a hole cut in the drape. And what you can't see is that the male's penis is coming out through the hole. This helper here, which I believe is Sierra, is holding the penis of the male just so it stays out and exposed through that hole. And then this other helper here is holding just a clean, new plastic disposable cup in front of the penis, so that when the ejaculate comes out, when the sperm comes out, it goes into this clean plastic cup. And I am here with the probe in this hand. In my other hand, this is the unit that generates the electricity. And so I'm gently sending pulses through one right after another, up and down, the pulses until there's an ejaculation. And then we keep doing that until the sample that comes out stops being cloudy, which indicates the presence of sperm, and is purely clear, which shows that we're just getting prostatic fluid and no more sperm in the prostatic fluid. And when that happens, we stop the procedure. We take the party cups over to the microscope, and then I do the rest of the work there. And essentially that work is confirming that we have sperm, confirming that it's good enough quality, there are enough sperm, and that they're swimming and looking good to do the freeze process. And the freeze process takes a couple hours. It's this process of slowly cooling down the sperm while it's mixed with special kind of media that protect the sperm during the process, buffers and sugar solutions that help protect the sperm as it cools down. And then eventually, once it gets down to about refrigerator temperatures, then we mix it with another special kind of media that has ingredients like glycerol that help to pull the water through osmosis out of the sperm cells, so that when they actually are frozen, you don't have ice crystals forming inside of the sperm cells, which would damage the little organelles and damage the DNA. So we pull the water out. So in essence, we're dehydrating the cells, and then we put them over liquid nitrogen vapor for about 10 or 15 minutes, and then we plunge them into the liquid nitrogen. And at that point, they're within plastic straws. So we draw them up into plastic straws, and the plastic straws are plunged into the liquid nitrogen, and the sperm is frozen inside of those straws. The straws are sealed on both ends. So then they can be stored in the liquid nitrogen in those straws for essentially decades until we want to use them, at which point we can thaw them back up to body temperature and then artificially inseminate females. So these straws are stored in liquid nitrogen tanks. The ones for the Mexican gray wolves are all stored at the St. Louis Zoo in Missouri, and they keep the entire bank there. So let's talk about a history of artificial insemination in gray wolves. In the early 2000s, a number of gray wolves were inseminated artificially and had variable success with having puppies. This was not with frozen semen. All of this was with fresh semen. So semen was collected from a male using electroejaculation and then immediately turned around and used to breed the female. So no cooling or freezing was used. In 2003, they tried this in 3 females, 2 of which were subsequently bred naturally. All of them had pups. So really, we could only say definitively that one of them was a true success because it was only bred with artificial insemination. In 2004, 3 females, different females, were again bred. One of them was successful. In 2005, 3 females were again bred with fresh semen using a different technique that gets the sperm all the way up in the uterus. And all 3 of them carried the puppies to term. One of them died during C-section complication. So that was unfortunate, but at least the proof of the concept was still there, with pregnancy working really well. So we know that artificial insemination works well. Then in 2011, the first attempt was made with the frozen thawed semen, and it failed. There was no pregnancy. In 2014, we had 1 female bred with fresh semen, successful, and since then, up till 2017, there were no more attempts and no more successes with frozen thawed semen. So we tackled this. This is about when I came in and started helping with the project. And we began to look carefully at the timing. We're not going to spend a lot of time here on this slide, but I just wanted to point out that it's a bit of a complicated situation to figure out when a female wolf is fertile. We can, in domestic dogs, we can track estrogen as it goes up and down, and we can track progesterone as it rises. And we can look for this other hormone called LH when it surges. And using how these 3 hormones interact, we can determine when she ovulates and when her fertile period is after that. Now, canines are a bit tricky compared to other species because if we were here talking about any other species that we're largely familiar with, whether it's cattle or horses or sheep or goats or pigs or cats or humans for that matter, whenever females of those species ovulate, the eggs are immediately fertile at the time of ovulation and ready to fuse with a sperm cell and form a little conceptus, little embryo. And the eggs are only fertile after ovulation for a few hours. And if they don't fuse with a sperm cell in that short period of time, they'll die, and you have to wait till the next ovulation, whenever that is for that species or that female, for another chance at pregnancy. So with women, it would be roughly once a month. With a wolf, or it's once a year that they ovulate. And other differences are that when canines in general, domestic dogs and wolves, when they ovulate, those eggs are not fertile yet. It takes a couple more days after ovulation, while the eggs are waiting in the oviducts, for them to mature and become at a stage where they can be fertilized. And then they're not just fertile for a few hours, they're fertile for a few days. So there's a lot of differences when you're dealing with dogs compared to other species, and we have to juggle that. In domestic animals, we can draw blood every 2 or 3 days and track this rise in progesterone, and that helps us determine when they're ovulating, but it requires blood draws every 2 or 3 days. So that's problematic with a wolf, because it requires going in and catching the wolf every 2 or 3 days, which, while it can be done very proficiently and quickly, is always still a bit of a stressful event for the wolf. And if you only have to do it once, that's one thing. But if you're doing it every 2 or 3 days, it does begin to raise the question of is the stress going to be causing problems with the wolf as far as its general health and fertility and ability to carry a pregnancy. So we're handicapped to in that respect when we're tracking the cycle in a wolf. Another thing we can use in domestic dogs is we can take little swabs of their vaginal cells and look at them under the microscope and see how they change throughout the cycle, and that helps us. But again, with a wolf, that would require a capture. So the only thing that we have that's not stressful or invasive with a wolf to track the cycle is fecal hormones. We can collect their droppings and analyze the feces there, but that's a problem because most of these wolves are in multi animal packs, and so you'd have to have somebody watching and noticing when that wolf defecates and going to get the poop just from that wolf defecating. And it takes a lot longer to get those results. It takes days, which is a problem. And the exactness of what the levels are also not exact enough, because what's in the poop is an average from the body. It's like the body brings it all together through the liver, through the kidneys, through the liver, and pushes that out into the feces. And it's an average of over since the last time the wolf ate, whereas the blood is an instantaneous measure of what the hormone is. So my point of all of this is not to complicate things too much for you, but just to illustrate it is complicated, and it's not easy to track exactly when the wolf is fertile. So the breeding season in wolves, as I've mentioned, they just cycle once a year. Domestic dogs usually cycle about twice a year, but their ancestors, the wolf, once a year. And it's usually in late January, it can go to as much as early April, but usually it's like mid-February to mid-March is the peak time of cycling and fertility for wolves. The wolf is not fertile for that whole month. Each individual wolf will be fertile for a few days within that time period. And so trying to nail down when this specific wolf is fertile and therefore would be a good time for artificial insemination with frozen semen is the trick. And I'm going to talk about how we try to have done that. So trying to predict that time when it happens naturally, as I've just explained, is difficult. And it either involves lots of stressful interventions with the female, or you could watch for when she's showing pairing activity with a male. But the problem there is if she breeds and the male is fertile, then she's probably going to have puppies from that male, and you want her to have puppies from the frozen semen. So that doesn't work very well. Sometimes we've tried to put the female in an adjacent pen so we can just see when she's displaying to the male and use that as a time to get close to narrowing it down. And that's what we've done in sometimes in the past. But this crew that it takes to get this all done is quite big and spread out throughout the United States. So if you've got a female, for example, if we were breeding one of the females at California Wolf Center, you've got the crew there that is able to watch the females and look at behavior and alert the rest of us when it seems like she's ready. But then we've got just a couple days to try to arrange somebody like me to come in to do the insemination, and somebody like a veterinarian and technicians to come in and be able to do the anesthesia. And you'd have to have people in St. Louis ready to ship the semen out. And that can be done ahead of time, but not too much ahead of time, because it has to stay in a liquid nitrogen specialized tank. So there's a lot of moving parts. And most of us have other jobs. I have a clinic that I'm running, and the local veterinarian has a clinic that they're running, the one that would do anesthesia. And so being able to just drop everything in 1 or 2 days notice and leave is tight, and that's difficult. So what we've tried instead is to do estrus synchronization, where we induce the female to come into heat in a time period that we can then prepare for ahead of time with a little heads up. And we do this by mimicking what the natural cycle does. So normally, like you see here on the right side of this diagram, hypothalamus, that's in the brain. Normally, it secretes this hormone called GnRH, gonadotropin releasing hormone. And that goes to another part of the brain, the anterior pituitary, and stimulates it to release a couple hormones, LH and FSH, which then go to the gonads, so the ovaries in a female. And FSH stimulates the follicle development, and LH stimulates ovulation. So if we then use an implant that has a drug that works like GnRH, and we can place that under the wolf's skin, it will then start that process on a time frame that we dictate. And there are a couple options for this drug. Ovuplant was an older one. A newer one is called Suprelorin. Doesn't really matter. The important point is we have options for a drug that works just like this GnRH. So we would give that drug 10 days before we expect her to come into heat, or we're inducing her to come into heat. So we'll do it a little ahead of time in the season, because we don't want her to come in on her own on an unpredictable schedule. So we'll give this implant, and then about 10 days later, we'll start monitoring her more closely. And that's what we've done in the past. So we started evaluating about 10 to 14 days after we put the implant in. And we found that seems to work with the rise in progesterone. If we do it too early, if we start monitoring too early after the implant goes in, then we're doing multiples of these captures, which even if you've got a very experienced crew, and even if you've got a relatively complacent wolf, it still is a stressful event for the wolf. And so we want to minimize the number of times that we have to do that. So we wait. But we don't want to wait too long, because if we wait too long, then we miss her ovulation, we miss the fertile period, and the whole thing was for nothing. So the trick was to try to figure out what's the sweet spot where we only have to do 1 or 2 captures before it's time to do the actual insemination. And it seems like we thought we had that dialed in. And then once we get the wolf in and we do the insemination, we stick an endoscope. So the wolf is anesthetized, and we stick an endoscope into the vaginal cavity. And that's what you're seeing here on the screen. It's a bit blurry because it's moist in there and there's condensation on the lens, but those pink folds that you see going past are the vaginal folds. And with it being as folded as it is, that's what happens when a female canine is in heat. So here you can actually see the procedure. Okay, here you can actually still see the procedure. And that's the endoscope I'm holding in my hand. It's inserted into the vagina, and I'm looking at a screen there. This is what's on the screen. So I'm getting close to the cervix. I want to go slow because I want to make sure that I am gentle and careful, that I'm just following the natural path of the vagina and not scraping along the wall. And then eventually it gets to the end of the vagina, which is about here. And then at that point, I back up a little bit and I look around to see if I can see the cervix. And that is the cervix right here. And this is the entrance to the cervix right there. So at that point, then we introduce a catheter into the endoscope. It passes all the, there's the catheter pushing in there now. You can see that right here's the catheter. And so the trick now is to try to manipulate that so it goes right into the opening of the cervix. And that can be tricky because the cervix is usually not a straight shot. It points downwards. And so you've got to be able to somehow angle it up to go in. This is all in real time. So this whole video is about 4 minutes long. So you can appreciate about how long this takes. It doesn't take that long to do, but it seems like a while when you're watching it. Now the catheter is just below the opening of the cervix. I'm backing it up, and there it just popped into the opening. And now I advance the catheter. It's getting stuck on a fold, so I have to wiggle it around, just gently twist it and change the angle a little bit. And then you'll see it will advance really nicely forward through the cervix. There it goes. And then the tip of that will be in the uterus. So at that point, we then connect outside, which it'll switch to in a second here, the video. We connect a syringe that has the thawed semen in it. So the semen's been taken out of the liquid nitrogen. It's been brought up to body temperature, and quick evaluation done to make sure that it is living. And then here we are gently and slowly injecting that into this catheter that is going through that endoscope, through the cervix and into the uterus. The liquid is yellow because actually egg yolk is one of the ingredients to the extender. Here's what it looked like under the microscope. So you can see we have lots of swimming sperm there. Some of the sperm are agglutinated to each other, but a lot of them are doing the right thing. And then on the screen, you'll see this bubbling as air that was in the catheter comes back out. And that's pretty much it. And then you pull the endoscope out, just pull it straight out and wake the wolf up. And that's the whole process. So the trick is really the timing. So we did this in 2017, and to a number of, I think we did 4 wolves. And we had, of those, one of them got pregnant. And sorry, 3 wolves, and one of them got pregnant. And she had a singleton, which is what the picture of this wolf is here. So it was success. Here was a wolf pup that was produced with just frozen semen from a male that was long deceased. So its genetic value was huge. But when you consider that wolf pup litters are usually 4 to 6 pups, and we had 1, and we bred 3 females, and usually if you have 3 females bred naturally, you'd get most of the time 3 pregnancies. The success rate was quite low. And in subsequent years since 2017, we haven't been able to even repeat that. So where we're at now is really trying to figure out how do we get this timing down. And at this point this year, we've decided to back off of the synchronization for a while and try to actually follow a natural cycle, and have to have people do that drop of a hat, fly into a place to breed, to do artificial insemination on a female when she's naturally ready. We've talked about using vasectomized males, so intact males, but that have their ducts, like the same thing that happens to a man when he's vasectomized. You cut the tubes, and so the sperm can't get out. If you have a vasectomized male and with a female, they can breed, and hey, this has got to be the right time, or at least has very high likelihood of being the right time. She won't get pregnant because the male's vasectomized, and we can quickly bring our crew in to do the frozen semen insemination. That would involve only one capture of the female, so less stress, and hopefully would give us much more confidence that the whole technique was working. And then we could start backing up again and try to work on some synchronization protocols. So that's where we're at currently. This is a picture of that pup in 2017. So he's now a mature male, and he's one of the most valuable, genetically most valuable Mexican wolves in the population, because his father was a wolf that was deceased from years back. So I think I've pretty much talked about this stuff. And so where we can conclude where we're at is that we know that the frozen semen is viable. We did produce a pup. The TCI is actually pretty straightforward to do in field conditions. It doesn't have to be in a hospital like I have in my normal clinic. Timing I'm confident that timing is our problem, figuring out when, but I also think that stress is a confounding factor. Even if we get the timing right, I worry that if we've caused too much stress, we might be affecting the fertility of that cycle. So those are the discussions that are happening right now. But if this works, the reason why this is all very important is because it allows us to use genetics from males that have been deceased. It allows us to take genetics from current males and preserve them for the future. So when they are deceased and gone, their genetics may become more valuable in the future. We have those genetics that we can use. It allows us to get, we could actually, with just a little effort, get frozen semen from wild wolves and bring that semen into the captive population without having to bring an actual animal into the population. It also allows us to share genetics between Mexico and the United States quite easily with the frozen semen. So there are a lot of advantages to being able to use this application once we get the unknowns figured out. And that's all I have for today. So I'm happy to take any questions.
Thank you, Bruce. We'll just give a minute for people to be able to type in some questions.
I have a question. I'll start with one if you can hear me.
Yep.
Bruce, you mentioned you're moving to do or attempting to do more quick live artificial insemination for the next phase. Have you selected any facilities that you're going to be using for that, or have used that are part of the program right now with the SSP?
Okay, that's a really good question. So that decision is made every year. The wolves come into they cycle, have a breeding season every year. And that's in, as I said, January, February, March. So in the summertime, the wolf management groups meet, and every year they look at the current state of the wolf in captivity and in the wild and determine what the plan is for the upcoming year. They determine natural breeding pairs. They determine pairs that are overrepresented and shouldn't be breeding and might need to have contraception. They determine movement of wolves from one facility to another based on needs, both of the facilities and the genetics of the population. And we talk about artificial insemination. And so sometimes those decisions are made because you have a pair that are matched. They're a breeding pair, or maybe I shouldn't say breeding pair. They're a matched pair, an alpha male and alpha female. They've got a good relationship with each other, and there's no desire to split them up, but perhaps the female has valuable genetics and the male is overrepresented. And so that would be a decision where we perhaps separate the male and female just by a fence line during the breeding season, and we artificially inseminate the female with another male's genetics, and then we allow her partner back in, and he's none the wiser. He'll think the pups are his, and they can take care of them. So every year that decision changes as to where that's going to happen, and it's based off of genetics, and it's based off of the ability of the different facilities to handle the breedings and the subsequent pups. And one more factor actually that I should bring up nowadays, because we are talking about reintroduction. If you reintroduce an adult, or sorry, I'll say if you reintroduce an adult wolf, you need all sorts of permits to do that, depending on the state government and where they're at. There's all sorts of red tape and hoops you have to jump through to be able to get that approved. A loophole is that you can take puppies and put them from the captivity into the wild in a captive litter. And that is done with less requirements, more easily. So that's called cross fostering. You can also take puppies out of the wild and put them into a captive litter. As long as you've got a litter that's born in the captivity and born in the wild within a few days of each other, so the pups are roughly the same age, then you can do that. And so the rangers that are managing the wild Mexican gray wolves have a pretty good idea when those pups are born, because they've got radio collars on some of the females, and when the female's signal stops moving around, it indicates she's denned up and having her pups. So they can pretty accurately determine when the birthdays are for the wild litters. And of course, the captive litters we know because we have eyes on them. And so if we have two that match up, that's an opportunity for doing cross fostering. And we can then take pups out of captivity and put them in the wild and vice versa. And if there's a tight population with little room for more wolves, as we currently have in captivity right now, most of the spaces are filled, that can still be an opportunity for still having litters if we can synchronize them. Luckily, with a wild litter, we can take most of the pups and take them out and have them essentially be released into the wild by joining a wild litter, and not increase the population in captivity at a time when there's not a lot of space for that.
Thank
That was a
you.
long answer. The short answer is that every year they reassess.
Dr. Christensen, I have a question of someone who's interested in this type of veterinary medicine and is wondering what's the best way to start that career path.
Well, first you have to become a veterinarian. And so there's all of those steps to get there, which generally requires a bachelor's degree of some sort, and then doing the prereqs to get into vet school. Once you get into vet school, the most important thing is just to study hard and become a good general veterinarian. But after you graduate from vet school, generally you've got a year of internship. And then if you want to specialize in fertility or theriogenology, then you would do a residency for 2 or 3 years, which is specialty training in reproduction and fertility. Now, as far as getting involved in zoo stuff, that is somewhat opportunistic, but it's a lot of you exerting your interest and being willing to make yourself available. So you develop the skills that are marketable and useful, and then you go out and meet people. That's exactly what I did.
Okay, and then we have another question. I know this is done in zoo settings, etc., but is this type of procedure done with other species for reintroduction into the wild, or is it still relatively new?
Yeah, this has been done in a number of species. It's not done a lot, and I would say the impact in general is minimal. There are exceptions that have been well publicized that are really great, but it's good to keep in mind that a lot of effort goes into this, and they aren't all successes. But the California condor is a great example. That's a bird, obviously, so that is much different because we're not doing artificial insemination like this, and it's birds work entirely different than mammals. But in mammals, a really good example of a success story is the black-footed ferret, and that was a an example of artificial insemination and frozen semen being used to help bring back that species that was entirely extinct in the wild, much like the Mexican gray wolf, and was brought back through captive breeding programs and reintroduced to the wilds. There have been a number of examples of ungulate species, different antelope species. We've done work like this with bighorn sheep. So the short answer is yes, this has definitely been done in other species. Everything from ferrets to elephants.
Okay, this is going to be the last question, and I know a lot of people appreciated and said your presentation was easy to understand, so maybe that's why there's not that many questions. But have there been any adverse health or medical issues that occurred because of this procedure with the wolves you've done?
No. I do this on hundreds and hundreds of domestic dogs, so that's probably a better measure, because the bigger the numbers, the bigger the chance for there to be something going wrong, because we've done this on so few wolves compared to domestic dogs. And besides me, my colleagues have done this on thousands of domestic dogs. And doing artificial breeding, this is called TCI, transcervical insemination. It doesn't cause complications. The one you would think it might cause is a uterine infection, and we just don't see that. There's no higher rate of uterine infection after this procedure than just normal breeding or not breeding and just having a cycle. So the short answer is no. The one thing that could happen if you weren't careful is that very thin endoscope. If you were to push it blindly, you could poke right through the vagina into the abdomen. And I've never had that happen while doing a TCI. I have heard of that happening, and I've never heard of an animal having a problem, because it's like a small poke, and you put them on antibiotics generally if that happens, and they're fine. So no. The only problem I would be most concerned about is general anesthesia. There can always be a problem with general anesthesia. The risk for that is low, but it's there.
Thank you so much, Dr. Christensen. Was there anything that you wanted to add at the end before Teresa gives a quick announcement?
No, I'm good. Thank you so much for inviting me.
Thank you so
Thank
much.
you so much.
Great.
Amazing.
Sierra, do you just want to add a couple comments about our work with the reintroduction and cross-fostering?
Yeah, so as Bruce had kind of mentioned, right now with United States, it's very difficult to do adult releases. We actually don't do any adult releases right now in the US from captive facilities, but we do partake in cross-fostering, which is another awesome use potentially for this artificial insemination, where we can get wolf DNA that maybe has been dead for decades back out into the wild through cross-fostering, where puppies are placed into wild wolf dens and then raised with the wild wolf pack. So we've been able to cross-foster a couple times here this past year, or past month, because it was very recent. 16 puppies from all over the United States were cross-fostered into the wild. So really, really awesome. And hopefully we'll be able to do more with AI, and hopefully do more with cross-fostering. I think that'd be really wonderful for the genetic diversity of the wild populations.
Great. Thank you. So I want to thank Dr. Christensen for taking time away from his very important work to talk with us about assisted reproduction. I will say personally, having him at our center, I've never looked at party cups in quite the same way as the use of them. So that was just a side comment that was amusing for our day with Dr. Christensen at the California Wolf Center. So our next webinar is planned for early September, and we will feature one of our past interns at our center, and she's going to present her findings on a research project she conducted. It was an investigation on non-lethal deterrence methods in captive Northwestern wolves at the California Wolf Center. So stay tuned for details on the date of this webinar, and be sure to register. Thank you again for attending our webinar today. Come and see our wolves sometime soon. It may be warm up in Julian, but our wolves are still active, and we want to encourage you to come up and see them. And thank you again, Dr. Christensen and our team, for participating today. Have a great rest of your day and thanks again. Bye now.
Bye.